Welcome everyone. My name is Arnoud van Haarst, Director of Scientific Affairs at Celerion. It is my pleasure to introduce you to Celerion in this presentation. I will expand on our capabilities for exploratory clinical trials as a CRO, but in particular I will address how we have adapted our clinical operations to the COVID-19 environment. Celerion is a leading global provider of early clinical development services, which support a collection of clinical data in the early stage of drug development, enabling you to take critical go, no-go decisions on new drug candidates. We collaborate with companies of all sizes, with the majority being biotechs, and with just over 1,000 employees globally, Celerion is an ideal-sized partner for them. Celerion provides full services from study design, subject recruitment, trial conduct to reporting, as well as bioanalytical services and in-house clinical lab services. However, I want to focus on one of our key capabilities, which are our 3 dedicated phase 1 units in the US and in Europe, here marked with red bullets, adding up to a global capacity of over 600 beds. Obviously, in response to the COVID-19 pandemic, we had to adapt our operations drastically so as to allow safe conduct of clinical research and enable continuation of early stage drug development. Now let's first zoom in on our clinical research facility in Europe, in the UK. Our clinical research unit in the UK is based in Belfast and has been open ever since 1990. It is a MHRA accredited phase 1 unit, harboring 78 beds across 4 clinical wards, and is operated by over 100 staff. During the 30 years that we have been operational, we have completed over 1,000 studies in our Belfast unit. And over time, we have recruited from the local population, and our database currently counts over 25,000 participants, including patients. At our Belfast clinic, we run trials recruiting both healthy volunteers and patients. And typical examples of early stage trials for both new clinical entities and biologics or biosimilars are first-in-human studies and equivalent studies. But other exploratory trials may evaluate drug-drug interactions, effect of smoking, food effect, or pharmacokinetics of drugs in special populations such as obese and elderly. We have extensive experience with complex study designs such as umbrella protocols integrating the aforementioned aspects. Another example of a complex design, which is very familiar to us, are hybrid studies combining normal healthy volunteers and patient panels, which can be studied in-house. These are often referred to as proof of principle or proof of concept studies, and commonly require assessments of biomarkers. So to get a clue of whether a drug is working in humans as it was designed is key to applied translational medicine. Proof of principle studies often aim to provide early signals of clinical safety and efficacy, but may also address whether a drug reaches its target or whether a novel mechanism of action works out in practice. So to a certain extent, such information can be gathered in healthy volunteers, but mostly patients are required to study disease-related conditions or biomarkers. To get a good sense of proof of concept, one therefore needs small numbers of patients usually, preferably with stable disease, investigators to ensure scientific and medical robustness of the study, and a controlled study setting meeting all the requirements for early phase trials. Pulmonary medicine is a good example of an area with trials enrolling patients and involving various biomarkers as well as complex physiological assessments. And this is a field we have been very successful in. An important pillar of our success is our partnership with Queen's University Belfast. For instance, our close collaboration with Professor Stuart Elburn, former Dean of the School of Medicine, is focused on respiratory disorders such as asthma, COPD, and cystic fibrosis. Queen's University provides scientific expertise as well as access to patient populations, but the experts have also supported us to implement specialized procedures within our clinic. Celerion, on the other hand, contributes high-quality conduct of clinical studies with a strong ability to perform a wide range of techniques.
The collaboration between my research group, Celerion's clinic, and our combined staff provides the ideal platform for performing early clinical research to assess proof of concept for new drugs to treat asthma, COPD and cystic fibrosis.
In Belfast, we now are a leader in the conduct of respiratory clinical studies through this partnership with Queen's University. And the breadth of techniques we can offer in-house is also provided as a snapshot in this slide. So these technologies have been developed in collaboration with Queen's University and include, for instance, lung clearance index, bronchoalveolar lavage, challenge models, body plethysmography, fractional exhaled nitric oxide, and spirometry. As alluded to earlier, apart from healthy volunteer studies, we have extensive experience with the evaluation of patient populations in-house. One particular example I want to raise here are studies enrolling patients with cystic fibrosis, because this experience has been very instrumental for dealing with the current COVID-19 pandemic. So since lung physiology of cystic fibrosis patients is so dissimilar from healthy lungs, they may experience drug tolerability issues and also display pharmacokinetic characteristics very different from healthy subjects, necessitating exploratory testing in patients. However, exploratory studies in cystic fibrosis not only face the usual challenge for early patient trials, such as co-medications, comorbidities, a lack of therapeutic benefit, etc., but on top of that, cystic fibrosis is a rare disease, further complicating recruitment. And assessment of drug effects in the lung often requires complex methodologies, if at all feasible in this population. But above all, cystic fibrosis patients have a high risk of cross-infecting one another, which demands special measures during in-house study management. So to that purpose, we have adapted standard operating procedures in our clinic, for instance, to increase stringency of site access and cleaning policies, but also to safeguard patient isolation and infection prevention. And these routines have helped us adapt our operations in the current situation. So in response to the COVID-19 pandemic, we have drawn up risk mitigation plans for our clinical research facilities and incorporated measures for clinical operations to screen, clean and isolate for COVID-19 virus during trial operations, in line with MHRA and FDA guidelines and local regulations, etc. These have also been laid down in SOPs that are instrumental in training our staff. With regard to social distancing, isolation and distancing procedures are now in place, and we have segregation in the clinic, also allowing to isolate subjects in case of suspected infections. All participants, staff and visitors will have their temperature checked and be queried about COVID-19 symptoms or contact with positive patients upon entering Celerion facilities. And access will be denied to anyone showing symptoms of the disease. We have implemented regular COVID-19 testing on site for participants and staff, in particular PCR screening for virus detection. And staff will be tested at least every 30 days. Participants and staff will wear appropriate personal protective equipment, PPE, during screening visits and while confined at our clinics. So, for instance, masks and overshoes. This will be event and study specific. So, for instance, full PPE will be worn when taking swabs for COVID-19 testing. Amplified cleaning measures have been put in place with special attention paid to high-touch areas. Regular hand washing is encouraged for all participants and staff, and hand sanitizer is available at facility entry points and throughout the building. For study participants that are to be screened for eligibility, we follow a three-tiered COVID-19 screening process. So first, we will arrange a call and query subjects for symptoms. Second, during a screening visit, symptoms will be queried and temperature will be checked, and a nasopharyngeal swab will be taken for COVID-19 PCR testing. And finally, the same tests will be taken when subjects check in for the actual study visit and subsequent confinement. These adapted procedures, however, may require longer confinement periods, as subjects may need to remain in isolation until lab results are available. Now, during the pandemic, there have been restrictions in particular towards lung function tests because of their risk of spreading SARS-CoV-2 virus via aerosols during test procedures. The European Respiratory Society, ERS, advised that test procedures for clinical evaluation of patients be limited primarily to spirometry and diffusing capacity, while other tests were only to be introduced when risk had been appropriately evaluated. Lung volumes by whole body plethysmography have also been feasible, provided that droplet contamination could be contained. The ERS also stated that local prevalence of the virus should dictate the level of safety precautions, while distinguishing 3 levels of safety recommendations, being pandemic, post-peak phase, and post-pandemic, each requiring specific restrictions and PPEs. Full operations of lung function services may only resume when viral prevalence is low and reliable testing is in place. In general, testing of patients for routine care is slowly being resumed, guided by the safety level. For exploratory trials, timing of lung function tests around strict dosing times in combination with frequent blood sampling usually demand that such procedures are performed on site. As mentioned earlier, the type of tests and procedures determine what kind of PPE should be used, in line with their potential for aerosol generation. And Celerion has resumed enrolling healthy volunteers and the performance of lung function testing in its Belfast clinic, as witnessed on this slide also. The conduct of test procedures is according to very strict safety measures. And along the same safety precautions, evaluation of patients as part of clinical trials can gradually be pursued. In Northern Ireland, the spread of SARS-CoV-2 has been well contained, and as a consequence, the prevalence of COVID-19 has been at one of the lowest rates within the UK, which facilitates resuming lung function testing in our Belfast clinic. SARS-CoV-2 may linger around for many years and resurge occasionally. Pandemic-related measures will probably remain in place to some extent, so things will only gradually return to a new normal. Implementation of SARS-CoV-2 viral screening is somewhat reminiscent of how HIV screening has become a routine test as part of entry criteria ever since the virus emerged in the 80s, and likewise COVID-19 screening may be there to stay as well. The level at which measures will be sustained will be dictated by local prevalence of the virus, by study procedures and their risk of transfection, and by the susceptibility of the population under study. With that, I would like to conclude my presentation, but if you have any questions, please feel free to contact me by email listed on this slide. I'll be happy to address. Thank you for watching.